Can everybody hear me well? Thank you. Okay. The whole concept of my talk is about time. I guess like Stephen Hawkins likes to talk about time. What I noticed about life as a beekeeper was I was doing a lot of that for a long time. The results are that you lose productivity in your apiary, you don't realize a good honey crop, and there's a big chance your bees are going to go someplace they don't belong. Thankfully, I have a good managed area because I worked on a ladder company for a while as a fireman, and I know how to climb a ladder and catch the bees, but sometimes they do fly away. All right, so swarm prevention was talked about a little bit over time by New Jersey Beekeepers Association. But next, the timing procedure. For reference material concerning this, I want to direct you right off to Bee Source Points of View. Now, Kevin may have that linked on our website. But if you go to BeeSource, www.beesource.com, you go over to the left and go right down to the bottom and you see Walter Wright's name, and refer for this discussion and for Professor's discussion to the named articles. But in learning about Walter happened at one of these. And that is the valid reason for coming to a beekeepers meeting and also to support New Jersey Beekeepers Association, because our Beekeepers Association has everything that we do is really dedicated to the education of its beekeepers. And I also want to say special thanks to BeeSource for providing us with this excellent list of references and so forth, all about beekeeping and many different points of view, not just Walter Wright's. Checkerboarding procedure works to prevent swarming cues because it relieves the swarming stimuli, which result in an instinctive response. One thing we like to do with beehives is we like to relate human characteristics to them. We got to remember that they have a behavior. They're just honeybees, and they do what bees do, and they know what they're doing. Always keep in mind that honeybees know what they're doing. Timing of a checkerboard procedure is a crucial thing. Walter started out discussing this very early on in his articles that go back into the 90s. And one of the things that always comes over and over and over again in this conversation is the concept of the crucial nature of executing procedure in a very narrow window of time. Another thing that Walter does is he does an exquisite job of identifying what's blooming. So in all of his articles, practically all of them, you'll see he mentions either a month or more specifically in that month, what's blooming. So after I read the articles about 65 times, it dawned on me, what's blooming and when is it blooming? There's a clue. And how do you relate to that? Well, this is getting right to it. How do you relate to it? That took me a little bit of time because I'm a chemist and an engineer, but botany was in my background too. And so a long way back, and then a little while ago, I was studying insects, and we always noticed that, let's see now, we're talking about the timing in the insects. There's a relationship there, and that timing is called a life cycle. The life cycles are reoccurring things that happen over and over again from year to year to year. It turns out that that's the oldest science known to man, because that's how we got interested in the concept of time to start with. We noticed this with the food sources. We used to chase the geese and the elephants and whatever. And then when it comes to swarms, that's another thing. Timing the procedure requirements. Swarm dates from previous years. Why is the swarm date important? The swarm date is important because that is an indication of exactly when the event happens. It's your job to go fast forward and put your timing mechanism in, if you've got one or if you understand it. The timing mechanism that I was referring to with recurring events is grow degree days. And I'll just give you a definition of a grow degree day in just a minute. A local knowledge of grow degree days as a function of the day of the year. What that means is you can calculate, it's actually a measure of accumulated heat by the earth in your area. So that will locally affect what's there, what's growing, what's blooming, and what's hatching in the way of insects. Both plants and animals are affected by temperature like this. And you can measure this or reproduce it, go looking for that behavior by watching your grow degree days. Apply the procedure at the right time in early spring using the grow degree days experiment, and develop what records are records of what is blooming and when. And I purposely did not include pictures. Well, I didn't want to go yank things off the internet, because this is a little more complicated than that. And say, look, watch the forsythia or watch the maple, all right, because it might not be the right one, all right, and it might not bloom at the right time if I go yanking it off the internet. And to be honest with you, I didn't know this experiment was going to work, because I had to guess initially. And then what's even more complicated about this is this experiment really started the year before, because I had to run a control, and I had to see which is it. Is it space or is it time? And I'll talk about the space concept, about give them some space. That'll stop the swarming. And then the next thing I'll do is talk about the time. What happened? Okay.
Somebody's trying to hack.
Oh, is that what it is?
It says, please log into your...
Somebody get the hook? Oh, man. Oh, got it backwards. I shot myself, I guess. Here. It's not working. All right. In 2013, what I did essentially was I did an original experiment and read on, and this has nothing to really do with Walter Wright. It's about bottom supering for swarm prevention. And basically what I did was, I was looking at time when to do it early spring, because the guy said to do it then. So I trotted out to my bee yard with my drawn comb and my honey supers, and I went and I just mindlessly put them on. All of my beehives got honey supers. And so I let it go. Through the season I went, and sure enough, around May, about mid-May, late May, early June, I had my 8 swarms, and I successfully concluded that it could be about space, but it's not honey supers. There's something else going on there. All right, so that got me looking even further. The next year came around, but I had to get the bees through the year. But the next year comes along, and we went to a bee meeting and learned about Walter Wright stuff, and I started reading about that. Next. Okay, back again. Fast forward, after getting into Walter's literature and understanding exactly where the bee sequence is, what happens is, as the bees move in and out of late winter into spring, they're going after their brood nest, and they're laying eggs, and they're building volume of honeybees. They're increasing their numbers. In the forest, the honeybee is a forest creature. It evolved in the forest. Where are the bees? They're in a hollow tree. Out in my bee yard, they're in the hives. That is their home. And the other point is, where are the bees at this time of the year coming out of the winter? They're most of the time way up in the top, aren't they? That's a critical observation to make. Sometimes they're in the middle, but a lot of times they're in the middle and the top, more times than not. So the question we're asking is, can we use that swarm date and pop intervention in and do something at the right moment? And the experiment shows that we can actually do that perfectly well. Assume that the hive objectives begin as degree days get above 50 degrees. Now, I got to define for you what a degree day is, and a degree day is it's an average of the high temperature and the low temperature divided by 2. That's an average. So it's the high temperature, low temperature divided by 2 on a given day, minus a baseline. What's the baseline about? Well, below 50 degrees apparently, and this is what I learned by looking around for grow degree days as they relate to blooming plants that are honeybee plants, really don't do anything below 50 degrees. So that's nice because you get a 0. And the other thing I observed was that honeybees don't really do much either below 50 degrees. Or you can basically start looking in the hive at 50 degrees, but my experiment said that, and my experience said, this is a match. This is fine. So the application is heat, energy, warmth. And also, now we're bringing in, the plants are starting to react to this 50 degrees as well because it's early spring. And we know that our senses are telling us that the plants will start producing what? Nectar and pollen. So there, I chose an intervention date because what I did was I plotted the month and showed the grow degree days. And so what I did was I looked at my swarm date and subtracted 30, and there's just a little bit of temperature there. And I said, wow, that's interesting. And it's interesting because it's right at the beginning of spring. Right at the beginning of spring. It's almost like the bees know what they're doing. They want to get out there and start doing something right at the beginning of spring. So here is the earliest swarm date plotted, and there's your regular days of the year, and the first day of the year is April 1st, and over there it goes to April 30th and all into May. And then what happens is, this is grow degree days 50 as the baseline. Now, 50 degrees is arbitrary because we know that bees are active below 50 degrees and they will fly. I've seen them out this year, and I've also seen them bringing pollen back. So maybe it's okay to use 40 degrees, but you're always going to have a problem finding a correlation between bloom dates at 40 degrees. So your grow degree days 40 might be a little harder. The next thing is when to implement the procedure. You implement the procedure, well, what happens if your heating sequence for the seasons are whacked out? So what I had to do was take a gamble, because this was done in 2014, and what I did, got lucky again. The net result is, look at the temperature profile. This is really temperature and time. That's what it is. So then third box, and how I checkerboarded, and that's another story. How I checkerboarded was implemented on the 11th, on the, what day was this? The 13th day, and that was because I was scared that the 11th was a little cool. And guess what? That's another, that was me. It was a miscalculation by me because, you know what? A given day or two, the bees just went through the winter in their cold box. They're fine. I mean, they're good with temperature. It's almost impossible to start too early, you know, within a week or two, all right? So over here, notice that that's 0, and that means that the temperature on the average was below 50 degrees. Over here, as we get into April, this is good activity. The temperature on the average is above, and that's fine. How do I get this?
Back to the previous slide real quick.
Yeah, if I do it right.
So this is grow degree days.
Correct.
The flat line is below 50, and where it climbs is above 50. What does the arrow down say?
it says the earliest swarm of 2013 was day 41 of the season. And a reason why I identified 41 is because I decided I was going to subtract 30 from that number and just do it.
So on that day, which is day 41 of the season, you're saying 30 days back is when you take the activity to start swarm prevention methodology?
That's what that was my first guess.
Okay.
That was a calculated guess.
And the next slide, that arrow down, is this first day of the swarm?
This is when I actually did the procedure.
Okay, got it.
All right, and then I look around to see what's going on. I'm out, got my telescope looking up at the redbud, not redbud maple, by the way. The redbud maple is grow degrees 1 through 27. So a redbud maple is consistent, and this is what Walter does. This is what Walter does. He gives you a vegetation-rich description. But you've got to get the plants right. And I tell you what, if you don't identify the plants right, you're going to be led astray. Because how many different varieties of forsythia are there? Take forsythia, for example. We got lucky because I have some yellow blooming forsythia, and when I did the procedure, I'm looking around and hey, there's some yellow forsythia blooming. Now I'm excited. This is the apiary before I checkerboarded it, and going into the winter of 2013. And then in implementing the procedure, this is interesting because we give an idealistic presentation of how to checkerboard. It's not nearly so complicated as that, and the bees are so very forgiving. They took all of the above. So, in short order, what I did was I took, I had 2 of my hives did not survive the winter. So I had a good couple of boxes and a half of honey. The next thing was I took that honey and started all through the yard with it, and I parceled it out. So I parceled it out, and so I told you it's a little extra information than you really need, how I got my bees to go through the winter. But most of the bees were up in the top because it's a combination of bee candy and honey. So what happens is the cluster, they just go right up, and the bee candy provides a moisture sink too. So that's what I added that for. What I'm discovering more and more is the bees don't need the candy. If you're doing your hive management correctly, you're going through the winter on what God gave them, honey. The first best food. One hive of 14 was simply reversed. I took that one hive and did a simple reversal. That's a 2-box system. Your cluster's like this, and you clip the bottom off the cluster and you plop it on top. And what you've done is you have injected some space in there, and you have injected fresh comb. Some of the one hive got a perfect checkerboard. Most of the hives got 2 or 3 full frames of honey, but I checkerboarded it, tried to put it right in the center. Now that shuffle produces some interesting food for thought, because that means that the bees, they're looking more for space than not, but the honey helps entice them up. One thing that Walter said to me in a writing one time, I think I read this, that I actually think he talks to me, but brood comb and wax comb, the honeycomb, brood comb, honeycomb. Bees know the difference. Do not use honey supers for checkerboarding purposes. Use brood comb for this purpose, because the bees can tell the difference. Old black comb, get rid of it. You're better off using a fresh foundation with the checkerboarding maneuver than using the old black comb, because the old black comb doesn't work anymore. It's shrunk up. So use your foundation, and the bees will, guess what? They will fill it out for you, just like that. For the 14, okay, that goes through that, and we'll move along here. The next step is there's your bee candy, and the bees are happy there. We're still on this continuance of implementing the procedure. Okay, 6 frames. I talk about the empty comb, and the empty comb proved very valuable. And when I use Peerco, because I can see the eggs in there. And so here's the results of the procedure. The hives, every single one of them went right into, expanded into the 3rd box. I really couldn't tell the difference in quality between, now this is a 2-box group of bees that went into a 3rd full honey brood box. So it's 3 full brood boxes now. And then they went and migrated into that with absolutely no problem. It didn't matter if the foundation was fresh or if it was a little on the old side. They went to empty comb. They went to foundation with no choice, with no problem. I had fresh frame with Peerco, and they just jumped on it and filled it right out, just like it was an irritation. That's vaguely reminiscent of something. Somebody says when you inject something into, I think Michael Palmer, when you do a brood nest intrusion like this, the bees will view it as an irritation, and they will go and they will fill it out real quick. And that's one way to do this, all right? This is not a brood nest intrusion. You try to manipulate the brood nest as a unit, as a sphere, without disturbing it. That's the best part of this. So the unity and communication in that system is maintained intact. But then you turn around and you clip the bottom off because you got a Langstroth, right? So when you take the 2 boxes apart, you clip the bottom of the brood nest. So you've intruded on it because this is your tree trunk. That's the way it is. But that tells you something. The next step was a friend came over, Kevin came over, in mid-June for the purposes of evaluating hives for splits. It was remarkable, the quality of the bees and the brood shown in each hive. I have a good bunch of boxes this past year, and I had excellent production of bees. They were healthy, and one of the most remarkable remembrances is the total quality and quantity where the bees were. The bees were packed from top to bottom. So what does that tell you about congestion? Well, I know what Walter says. Walter says there's 2 kinds of congestion for the purposes of conversation. One kind of conversation is you got a lot of bees. But the other side of the conversation says you're impeding operation, because what happens is that's more or less the backfilling concept right there. So he had his mind on that as a cause for swarming. And so that is part of the process of the natural swarm or reproductive swarm idea, is backfilling and so forth. So the other thing is that the grow degree days seem to correlate with a checkerboarding date in different areas quite well. And the reason why that is because I went and I've been looking, and he has his tree blooms, and I take guesses at what that tree species actually is and calculate the grow degree days on it, and there it is. So just like the pebbles in a stream, the truth shines through. There are no signs of swarming in the yard in 2014, and I went from an annual swarm fest to looking around in the trees, well, where's the swarms? This is like the return of the swallows or the monarch butterflies. Every single year for years, it's an annual ritual. Me and my wife are out there chasing bees. So. And so this is the result right here. I now have these monoliths all over my backyard, and congestion is not an issue. Management of the honey became the issue. management of the honey, because that will impact on the hive as well. And so what I did was, the management technique would be called bottom supering, and that would just keep rotating them. And you maintain the height of the hive by firing up the honey super and keeping a box mature and empty, or in the process of being emptied. And so the net result is you will get your honey that way, and my production really showed it. And the stimulus behind this for Walter is that he is a producer of honey, quality honey in Tennessee. And he's his own R&D department, him and his family. What a group of people, that concept, and zeroed right in on the biology of this situation. And what they did was they walked through the life cycle, quick to grasp key elements of this, of the life cycle of the honeybee with respect to its reproductive biology, because the reproductive biology is fission, and it impacts dramatically on honey production. So this is my hives as they sit right now, with or without the snow. We got the blue painter's tape to cover the seams and so forth, and I have my bee candy up there as the moisture sink. And so now there's the food for thought. What has the observation of no swarms, what did that do to my life as a beekeeper? Oh wow, it just causes me to think more and more, because now I link up the observations about the reproductive biology end of it that's Walter has made, and now that I have a timing mechanism and a clock, I can accurately look into the hive and do what I'm supposed to do at the right moment to see what I'm supposed to see. This is the hypothesis right now for me, because now what I want to see is white wax. When do I walk out into the hive and see that white wax? I don't want to see a swarm, so I already know how to stop that. Then another thing is fill out my records. I have to ask y'all, what is a beekeeper without records? And what is a beekeeper without records if he doesn't know how to anticipate when to take those records? You have to know enough to know. That's a problem, and you got to know what you don't know. So the checkerboarding process and the nectar management. Now the nectar management, this is another dose of his subtlety. But once you grasp what he's talking about with this nectar management, it impacts directly on the biology of the hive as it applies to swarming. Because this congestion he's talking about is a buildup of nectar inside the brood nest. Stickies up the brood nest, but actually what is it showing? It shows you something. That hive has decided it is going to swarm. I believe they're leaving nectar behind for a time when the hive is going to go without a queen. There's a time lag, and I've noticed this more times than not, that the swarm leaves and behind is left what? Queen cells. The queen cells are incubating. So if you impact on this hive by ripping it all apart carelessly, you're going to kill the queen cells. Net result is you got a hive with no queen now. All right, so one of my next steps is do internal observations of hives and document the brood nest expansion using a timing mechanism. My timing mechanism is the grow degree days. Record swarm dates if they happen. This is what everybody should do. This is the importance of the swarm date, because you're never going to know which came first, the chicken or the egg, if you don't know how to measure when. I see the handout now. This is the handout, and what this turns out to be is a very most recent document. I'm not sure, but I asked Kevin about this document here. There's actually two references here, and what this does actually is give a level of credibility to my, I'll call this an observation, all right? Because we want to be scientists about this a little bit. We're beekeepers, A lot of us are farmers, and we all do a lot of other things besides. We're not practicing scientists. But here's what we got to do. We got to sift the reality out of this and test it and test it and test it and make sure that what we're doing is the right thing. So we want to talk about first establishing a credible timeline, and how does that relate to our time? The grow degree days relate to the trees' time. And who do the bees relate to? They got their own time, but they also do better with tree time than they do with wristwatches. All right, so we start in short order with the management of, there are two pieces of this. Both of these came from the Wright document. The first document was the summary of spring operations and what he does. And this is measured in Elkton, Tennessee tree time, which is on the border between Alabama and central Tennessee. And what across the top is your tree bloom. And now, you know, this is what I had to deal with when I looked at all of Walter's writings, is the tree bloom. Here it goes. Elm, maple, redbud, and for the long time I thought redbud was red maple, but it turns out redbud is his own tree. And then apple blossoms and black locust. Well, I have apples and black locusts as well, but probably not the same ones as him. So this becomes a little bit of a trick. But go with the flow. He threw in the calendar there, and so that is a good thing. All right, next, my swarm date, lucky shot, began the correlation process. So I know I got no swarms, and the question is why? So now this becomes a real experiment again, because out bubbles another observation. The day I chose was 30 days after the swarm date. But if we look down here, what I did was, it says new wax here and checkerboard here. Well, if you read the article, and that's what you got to do, Walter says that if you want to time a checkerboard maneuver or procedure, a proper time to do this is to go back 8 weeks once you start working white wax or observing white wax in your hives. Well, if you're observing white wax in your hives and you haven't seen any swarms, you're in the main flow. The honeybees evolutionarily will reproduce to get their bees out there before the main flow. Why would that be? To maximize pollen and nectar for what's left. The foundling hives. These babies, they're new beehives. Another thing, it just so happens that the grow degree day correlation for that is around 40 or maybe 50, between 40 and 50 grow degree days. And I was looking for agreement. I'm always looking for agreement. It turns out that on my chart, when I did this, I had to figure out whether I got to go backwards or forwards. Here we go back in time. Here's my checkerboarding event. Look at what happened. Isn't that wonderful? Bang. 50 grow degree days. And the survey says checkerboard around 38, 40, 45. See here? Right here on the first page. And that's not even on the checkerboard manipulation page. It gets better. On the checkerboard manipulation page, which is also in another one of his articles, what I did a better job of doing was getting the grow degree days down and comparing and showing actually that his swarming sequence down there is no different than it is here. Does that make sense to everybody? It should make sense, because what shifts is their growing season. They got a longer growing season. And there's another point that comes out about grow degree days. Grow degree days, once you don't work backwards to get, once the bees start seeing 50 degrees, the candle's lit and they're on the roll. You don't, just because you're sticking at 50 for a while, which you will do at Tennessee, in Tennessee a lot longer than you do here, it doesn't mean anything. The bees are growing, the colony's moving, and the train is leaving. So, you gotta move. Go ahead.
Touch on new wax starts, new wax.
That's a concept, yeah. That's another thing, new wax, okay? What's the question?
Well, what is it?
Oh,
Early capped honey? What is new wax?
well, what happens is, Randy Oliver says that you can actually, the bees will tell you when they're getting crowded, because you'll see new white wax. The easiest way to see new white wax is when you open up your inner lid and you see this new white wax in there. The bees are trying to tell you something. There's a lot, they need a super, a honey super. That's what that means. There's wax producers in there. Now, that's another conversation, but my purpose here is to try to tell you accurately time. But
Wait, so the point is, you'll literally see white wax?
yeah, you'll see it.
They'll be gluing it, they've got no place for it. They're gonna put it under the inner cover, sometimes they'll put it along the bottom of the bars.
All right, and then
You
another,
put that honey
It's white.
super on.
It's literally
You
white.
put it on, take the one that's filled up and put the new one in.
yes, that's bottom supering. Okay? And what you do is that's another discussion of supering. All right. And then another thing about this whole thing is the classes of bees. If they're going to swarm, you're going to have wax producers in the hive, an abundance of them. But if the hive decides something, and that's where we're going with this, the hive has made a decision. Classic decision. We're going to do something. We're going to swarm or we're going to stay. And if we stay, the population mix changes. There's not as many wax makers. This is brought out. You're going to have to observe this, but he talks about it in these papers. It's in the swarmers where there's wax makers. Why would there be wax makers in a swarm? Anybody got a solution for that one?
Gotta make a comb when you get to find a new home.
Got to build a home, a honeycomb, a place to live, a place to make new.
And wax makers are young bees, right?
Correct. Then that's consistent. There's so many things that are consistent about this. Is the age population and what we've seen before about the congestion levels and everything. But one thing is true. In my hives, they're very, very, very congested, but one thing I don't see is backfilling. I never saw any backfilling. And another thing, the numbers of supersede, I only saw queen cups, nascent queen cups. Never saw the classic supersedure cell, nice big one, in the middle of anywhere. That's another conversation, because each one of these little points you're talking about turns out to be a 1.5 hour talk. All right, so where are we at now? 5 minutes. Okay, getting to the, okay, let's take a look at this first page again. Go to the top where it says colony objective. The colony objective is to reproduce. The urge to reproduce is overwhelming. When I talk of swarms, there are basically two kinds of swarms. It is a reproductive swarm, and then a small subset is a housekeeping swarm or a fall swarm. That's a classic housekeeping swarm. But the fall swarm, like Barbara had, it could be a manifestation of the same things. And that could be an explanation as to why the hive would go through the effort to reproduce itself. So maybe a fall swarm is more like a reproductive swarm than it is a housekeeping. You can actually crowd the bees out of the hive, and that's why Walter calls them a housekeeping swarm, and it's on the beekeeper. It's a beekeeper mess up. I did that by taking a honey super and throwing it on a good hive and ruined it. The thing took off, literally, almost as quick as I did it. The next thing is the stepwise march to reproduction. Going, the idea to look on a line 4 where it says effects, and coming out of the winter, We're increasing brood nest volume by creating bees. We're creating a new worker force and so forth. And then as they hit the honey dome, that's a hard break. The hard break is there. They sit there literally looking at the roof of the inside of the beehive. And in the trees where they come from, they're being a forest creature, they're looking at where they started their nest a year or two ago. That's a hard break for them. They're going back, and there's a communication going on inside the hive. This is me thinking. Now they're going to do something about this. They're going to react to it. And then when I intervene by taking and doing a relief on their hard break at the right moment, I have done something there. It's like rebooting a computer or unplugging a power supply or something like that. The system has to start over again. This is me thinking. But one of the things we have to do now is understand this two ways. First, what's the practical application of this? And the beauty of the work is we're taking advantage of some kind of a response to a cue that's deeper than just, and it's that not just one. This is a whole hive organizing response. It's not just one little sting or whatever. It's not the bee responding to you scaring it and you're getting stung. This is a hive level decision that's going on here. This decision to stay. And if you can view beekeeping that way, that your hive will stay instead of leave if you let it or make it, then you'll be a better beekeeper and you will raise better bees for the purpose of honey. And the next step is the queens and so forth. You can make queens, or you can use these techniques that we're talking about here with a timing mechanism to manage your whole apiary in a better systematic way. That's the practical outcome of this. And go ahead, Doc.
Sorry, just to clarify, Charlie.
Yeah.
So you did your checkerboarding once.
Yes.
You didn't have to go in and do it again, right?
Oh, one thing I've left out, I did not let you, I didn't say, on my growth curves and everything, what I did show was that I added honey supers. I almost immediately added honey supers. To start with, I added one medium honey super with honey wax, not brood wax, it's honey wax. The comb. Correct. Now brood wax and honey wax, they're different. Got that. And guess what? I didn't get the intrusion. I didn't get bees growing up into my honey supers. And I just kept shuffling them and shuffling them. That's good. And then the next thing is, there's a lot of conversation that I could have about the dynamic of that yard and what I saw in that year. And how the bees would load a box, fill it up with honey, ripen the honey, and then they would go through it and load it up with brood. It is a marvelous thing to see, and there's no inkling of a swarm in sight. They're not going to do it. All you got to do is manage your honey. Questions?
When you say shuffling, are you talking about reversing the two bottom supers?
What did I just, related to what? When the bees, I was talking about what the bees do. They go through a process. When they lay their eggs, they plant or they produce a frame of honey. They'll ripen it, and they'll go into it, and they'll start laying eggs into it, and it's all classic brood nest observations. You're loading, you got a then you still see the solid cap of honey at the top. You see the pollen. You see the nearly full frames of brood, capped brood. And then that hive, and then the next one, and the next one, and the next one. Then they'll go down to the next box, the one below it, and they'll do that one. One of them, does that answer your question? This is not part of the checkerboarding. This is the bees doing their thing during the summer.
No, when you say shuffling, what is it that you do to shuffle?
Maybe start off with the reversal.
Oh, I got a reversed. All right, so we're talking about the checkerboarding or the reversal. View checkerboarding as a specialized case of a reversal. all right, where you've got, it's a location of the bees, and you're trying to give the bees a concept of space. The bees were in the first box during the summer. They're now at the top in the winter. Take the ones on the top, put it on the bottom, take the bottom box, and put it on the top. Just like that.
And you do this more than one time?
Nope, I do not do it but once. And then on top of that, I will put my honey supers.
Can I borrow your pointer?
Yes.
I mean, we followed Charlie when he did this. And also, Bob and I, how many were at the spring meeting last year at the mentoring hives where we did this? We did checkerboarding, right? So some of you saw this.
I see what you're
We'll
doing.
use the diagram here. Pretend that top box is not there, okay? That cover is on that, and now we're coming out of winter. You've got your cluster, it's growing, growing, growing. And you got a honey dome on the top, right? Honey dome on the top, this is a roof. What we did, the cluster grew, and it filled those two boxes. So to relieve pressure, we added a deep. See it?
Mm-hmm.
What'd they do? Now we're at the stage, what did they do? They filled it, right? Because the honey's coming. So now, you're at a point where you have a honey dome, and they filled the bottom brood chamber. What do? You checkerboard it. That's what this is.
Mm-hmm.
So you take that box, and you add another one, and you split its contents between the two. This brood chamber, typically a brood chamber will grow up to 3 boxes in New Jersey, right? So this brood chamber will expand now through this little bit and up into this box, and honey goes up here. So they'll eat this honey and make it brood. They'll eat it. That's their food while they're putting brood in there. They'll feed it to the young. And they'll pack that one with honey. So the way Charlie did this was he had 2 boxes, and on March 15th, which is 30 days back from his swarm dates from the previous year, March 15th last year, he put an extra box on to relieve pressure, and he did some checkerboarding. On April 1st, 1.5 weeks later, I think it was, Charlie?
No, it was really, the date was April the 13th. I did the checkerboarding, and then 1 week later, I threw on the honey super.
So that was the pattern. He gave space for the brood chamber to grow, and then when the nectar flow was full on, he gave space for the nectar. So it was a 2-part strategy, right? First one is make sure that you don't have the congestion in the brood chamber. Second part was give them adequate space to put honey reserves, right? And the key to it was
Nectar
taking
management.
his swarm dates from the previous year and mapping them to growth degree days. He was able to figure out backwards when 50 degrees hit, that's when I should take action. Right? Is that clear to everybody? And I don't know if that answers your question, Stan, but it should give you a concept of where Charlie went.
Go ahead.
Have you done it yet this year?
No, because it's 0 degree days, and I'm waiting for, I'm going to test it again, so I'm going to let it creep up to the beginning of April. And my objective this year, one of them is, we go over here, you see on the bottom line, I've got a date there in Bridgewater, and then up top is the time in Elkton, all right? The concept there was to show that the seasons are different there. But what remains the same? The grow degree days. So then if you drop it down, what I'm going to be doing is taking a look at some of the basic things that are happening here, but I don't want to mess up my honey crop or bust a queen cell unnecessarily. So what I want to do is take a look at white waxing, or look in the hive when backfilling should be happening. Do it like that to see if you're going to get a swarm. So you look for backfilling. If you see it, and I've seen it, you're going to get a swarm, most likely, because you're not really going to be able to turn that thing back. Or you can do more drastic stuff. That's what Walter says. Then you can go for brood nest intrusion. You can take and start banging things in there. That would be frames of empty comb right in the middle of this, and expand the brood nest this way, and pull queen cells if you think you can do it. And that's fine. People do that to raise queens. They go in there and harvest queen cells, so that's another application of this process here. But that's understanding what's going on when in the hive.
There's one more point to make to this. large brood, a large brood nest,
Yes.
gets a lot of honey. And that mentoring hive that we did last year, going into fall, it was heavy as could be because it had a huge workforce. When the nectar flow was on, which is key, right? One of the things that this does is it gives you an early workforce for when the nectar flow hits in earnest, and then they stack on a lot of food, and then they have food to get through the June, July, August dearth we have until you get the goldenrod in New Jersey, right? So it's about also sustainability in your colony. You don't have to feed them. And our mentoring hives went through the winter and came back out the other side, still heavy with excess stores and big clusters that made it through this brutally cold winter, right? So food for thought is following this. You know, we've learned here in New Jersey over the last couple of years that actually right now, March 15th and on, although it's been colder this year, Queen's in there doing their thing. As soon as they can get out and fly, they're hitting the skunk cabbage, they're hitting the snowdrops, they're hitting whatever else is out there right now, right? You're starting to see
Crocus.
a couple trees bloom. You've got purple flower, crocuses out,
Crocus.
right? So they're going right now. Now, don't get premature. It's 30 degrees out right now at night, right? So don't go home and crack your hives open. But what Charlie's saying is when we start consistently getting to those 50-degree days, you're going to want to start thinking about giving them the ability to have space to grow that cluster.
Okay, go ahead.
On your picture on the right with the 4 hives, so the 3 on the bottom are deeps?
Yeah, deep,
And then that's a medium super?
This is a deep because you pulled deep frames out. You could put a medium here. You don't have to use deeps. I just use it because that's the artwork I had for the picture. Your third box over here can be a medium. And there's no problem with brood being there, although some people don't like brood in their honey boxes.
Well, that's what Walter
They don't
says.
want to exchange them, right? So you have to think about this before you get started and have the right equipment on.
All right, but one thing I'm going to tell you is right now, in this, using this picture here, right here's where your bees are, most likely. I would say with 95% certainty your bees are in this vicinity. So what am I going to do with this system? I'm going to take the top 2, drop it straight down, take the bottom, and fish around in here for some spare honey. Remember what I told you. You don't need to have just absolute ideal even, you know, checkerboarding system. You got to have something to get the bees up there, and that's the idea. And in here there will be something. Is the system going to be perfect? This is a Langstroth hive. It's not a substitute for a tree. But one thing you can do, and this is another conversation, is put a little frame on the bottom. Put a shallow. Make that at the very bottom, turn it into a pollen pot. And let them fill it up with pollen all year long and leave it where it is. And what Walter does, when you read the papers, this will tell you, he uses that as another source of empty frames for the purposes of checkerboarding, okay? But did this befuddle you, or did I leave you in that? Because I went really fast with that.
Yeah, it's going to take me a little time.
Yeah.
That
Okay.
box could also be brood comb.
Yes.
It could be brood. You could put if you have a box that had brood comb in it, you put brood in there. They'll store brood and honey. But remember, you're trying to build a 3D brood chamber, right? So if you put your honey boxes in there, they're going to put brood in it, likely.
You want to break that honey dome if you're doing a simple reversal, separate it, you know, out. And then put honeycomb on top of that. If you put the honeycomb there, chances are it'll stay as honey.
So let's go through a couple more questions, and then I want to...
The top 2, you have your dark frames and your light frames.
Yep.
The dark one is a full frame of honey, and the
Yes.
light one is a foundation, maybe?
It can be foundation. So that's a really interesting question, right? Bees like to, when you have a brood comb and a brood comb, and you put a foundation in the middle, they don't like foundation next to brood. So they'll build that out. When you have a honeycomb, and you put something in the middle that's foundation, you could just roll the dice, right? And the reason for that is sometimes they'll take the 2 honeycomb ones and build them into the space. And sometimes they'll build honeycomb in there and build you a honeycomb frame. It doesn't, you have to just let it go, right? When they're in a wax building mode though, right, they have to have nectar flow to build wax. And they have to have young bees to build wax. And when you have nectar flow and young bees, you can put cardboard in there and they'll build on it, right? So chances are you should be okay with them building out honeycomb in between. Oh, look, I changed it. I didn't even know I did
That's
that.
brood nest opening, right?
Yeah, so this is relieving the brood chamber.
That's a
And
different
This
thing.
is the key thing. If you are pulling stuff, like this is, you're gonna do a split or something. When you're pulling things out of the brood chamber, try and give them drawn comb back. If you're in a wax building mode, go ahead and put foundation in. But that wasn't what we were here to talk about, so that's another technology.
One other thing about a big hive, and always remember this, what is the key characteristic of this because you didn't swarm? Answer, population density. How did it get that way? Bee reproducing, bee production. What goes along with honeybees in production? Brood cycles. And what happens with brood cycles? Jim?
Mites.
Mites.
Good point. This is a quick slide that you could get through. There's been a lot of conversation over the years, ever since I've been a beekeeper, about this is what you do in the spring. Your bees are up under the lid, you put them down below, and the brood chamber will grow because bees go up. I think that's a misnomer. Bees go where space is, but that's the conventional wisdom. If you have bees in the brood, and they got a honey dome on it, and you do that switch and you got honey over it, they may not necessarily move up. And whatever brood was in the top of this bottom box is now up here isolated away from that one. So you really kind of want them up, and you don't want that much heavy honey over top of them if you're going to do this brood reversal. You're better off, leave that box alone. They will build back down into the bottom one, okay?
Okay, but one thing, Walter addresses that very point almost right to the diagram in his most recent papers. And when he said, when you take and clip the brood, he said, now as long as you've got the bees, when you've got the bees,
Yeah.
okay, you can do this. But if you don't have the bees, most likely that hive is not going to swarm anyway, so leave it alone.
The problem with this is on a cold night, who's keeping them warm?
That's right.
On a cold night, who's keeping them warm? Okay, I want to say thank you to Charlie and to David, thank you.
Wow.
I wanted to move along because we got one more thing to do